Excellent. So thank you again, Paul, for that lovely introduction. I would just like to begin by acknowledging the traditional owners of the land in which all of us are located today, and pay my respects to their elders, past, present, and emerging. As a UQ ally, I would also like to acknowledge that Sunday was International Day against homophobia, biphobia, interphobia, and transphobia as well. So as Paul mentioned, I did my undergraduate degree at UCL in human genetics. And to be honest, it didn't involve as many humans as I was hoping it was going to. And so after a particularly punishing prac in my second year, in where I killed more Drosophila than I cared to mention, I went for a walk to clear my head, and I found myself in front of Greater Ormond Street Hospital for Sick Children. And I had one of those epiphanies where I thought, yes, this is what I'm supposed to be doing. I'm supposed to be involved in humans. So I reached out to the director of the genetics department, Professor Robin Winter, and asked to meet with him. And he's a huge influence in my life, and he hired me, gave me a summer studentship, and then allowed me to do my honors project with him and hired me as a research assistant for a year after I graduated. So, and during that time, I got to work with Maria Bittner-Glinzich, and it became very clear to me during that time that whatever I did, I wanted it to be clinical in focus, which is why I applied to do my Master's of Genetic Counselling at the University of Manchester. And during this time, I met Paul, who many of you will know. Yes, can you hear me?
Aideen, sorry, could I just interrupt you?
Yeah.
You've got it on, it's like the speaker mode. I don't know how you shifted onto the audience mode.
Oh,
if you
okay,
can do that.
Sorry. Okay, one of my
So we're seeing
you're seeing
your next slide, and
you're seeing. I'm spoiler alert. Spoiler alert.
Yes, exactly.
Okay.
Yeah.
Okay. All right. Let me. I don't quite know why it's doing that. When I did it, I wonder. View. I don't know how to take it off. Let's see. Undo.
Well, that's the
hide presenter
that's the
view.
same.
How about that? Okay. Does that work? No. No. Let's see again.
How about F5 on the keyboard?
Okay, hang on 1 second.
I think F5 brings up the standard view.
I'll give that a shot. Function F5, I presume. There we go. Yeah. If I try doing something again, hold on a second. Sorry, guys. So what are you seeing now? Just my normal view?
Suggestion was to try F5 if that switches the view.
I'm trying to do that, but for some reason it's not cooperating. I wonder why.
It thinks you're plugged into another projector, I think.
It's because I've got 3 screens on the go here. So one of my screens, it's showing normally. Hold on a second. Let me stop this share for 1 second, guys, and I will. What I might do is I might put that on there, and then I might go to. So if you don't mind seeing my side profile instead of direct on, then I think I should be able to share that screen. Does that work? Please.
Yes.
Okay, good. Okay. All right. So sorry about that. So all right. So so I met Paul, did my master's and at that time, Paul was doing his postdoc in cold atomic physics at UCL. So most of you will know Paul as being head of bioinformatics for the Australian Translational Genomics Centre, but he wasn't into genetics before he met me. But he got a postdoc in Maryland. And so when I finished my master's, I applied for various positions and was lucky enough to get one, as Paul said, at the National Institutes of Health and the Genome Institute there. Barb Biesecker hired me to work with her on research and also to be the Associate Director of the joint NHGRI Johns Hopkins Masters of Genetic Counselling. I also worked with Bob Nussbaum in the genetics of Parkinson's disease research. So in a little bit more detail, and I'm just giving you a quick helicopter view to get a sense for what kind of research I do, because it's a little bit different, as Paul said, from the average here at Diamantina. So my psychosocial research was both quantitative and qualitative. So quantitative, we use validated measures to assess the impact of BRCA1 and 2 testing in families with hereditary breast and ovarian cancer. Looking at, and then we did qualitative research in a wide variety of studies, but one of the biggest of which was interviewing girls and women with Turner syndrome. And for those who don't know, Turner syndrome is caused by one X chromosome instead of 2. We did a lot about patient education and health decision tools. So when BRCA1 and 2 testing became available, a lot of women who had not a particularly strong family history were anxious about it and the possibility that they might carry the gene. So we recognized it would be a benefit in being able to educate people about this in a straightforward way. So using interactive computer programs to educate people. I also contributed to gene discovery studies, identifying families with inherited Parkinson's disease in my work with Bob, and wrote quite a few papers on genetic testing in Parkinson's disease with some guidelines, etc. Our research also involved clinical research, natural history studies in Proteus syndrome and others, and also doing fluorodopa scanning in individuals who had LRRK2 mutations in the family with inherited Parkinson's disease, looking to see if whether we could see kind of a positive PET scan in an individual who was at risk for developing Parkinson's disease, but it was as yet asymptomatic. After our second child was born, we decided we'd like to be closer to someone's family, so we moved back to Brisbane. And as Paul said, I took a clinical position at a specialized obstetrics and gynecology imaging practice in South Bank and worked there for 4 years. And while I was there, I was also very heavily involved in the Masters of Genetic Counselling program at Griffith, doing a lot of teaching and supervising master's students in their research projects. And that's actually how I first met Tatiane. I was also acting convener of that course while the then convener, Madeline Peterson, was writing up her thesis. So once Clodagh started school, my youngest started school, I decided I'd like to get back into research, and Paul was working for Matt at that stage. So I wrote to Matt, told him about my background and experience, and he hired me to be a study coordinator for a skeletal dysplasia study. And one day Paul was bemoaning the fact that no one was analyzing this exome sequencing data that he was generating from the skeletal dysplasia study. And I asked him whether he thought he could teach me how to do it. And he said, yeah, of course I can. So he taught me how to analyze the data, and I did. And in a very short period of time, I identified 3 new genes for short rib polydactyly. And it's hard to describe what a Sherlock Holmes frisson that generated. It was very, very exciting, especially because I knew what the value of a diagnosis would be for these families. So we were lucky to be able to reach out to Carol Wicking, who was working at IMB at the time, was a specialist in ciliopathy disorders, collaborate with her, and she did the functional work to back up what we were finding from the exome analysis, and we produced some papers that I'm really proud of. So Matt persuaded me that I could do a PhD without having to go into a lab, and that was important to me because I'm not a lab person. So my PhD was predominantly on the analysis of whole exome sequencing data. So I carried on doing gene discovery for a variety of skeletal dysplasias, and also for a skeletal dysplasia called Fryns syndrome. That is a rare disorder, but it's one of the, it's the most common cause of syndromic congenital diaphragmatic hernia. So for families who are diagnosed with a baby diagnosed with congenital diaphragmatic hernia, having this kind of information and prenatal diagnosis is really helpful. And all of the genes that I discovered were very, very quickly incorporated into clinical testing panels, which was really gratifying. I also analyzed data to identify modifiers. So these were genetic variants that were either in the same region, so they were allelic to the causative variant, or they were in related genes in the same pathway, which appear to modify the severity of the disease in those cases. When whole exome sequencing was first introduced, there was a lot of excitement about its potential from a gene discovery point of view, but there was a lot of cynicism about whether or not it could be used for diagnostic purposes. Predominantly because, unlike the name implies, whole exome sequencing, it wasn't whole. It wasn't able to successfully target and capture every exon in the exome. And so therefore people were concerned about its value. But the capture improved exponentially in the course of a very short period of time. So in the course of my PhD, I was able to write 3 papers on 3 different groups of disorders showing that whole exome sequencing was sensitive, specific, and cost-effective for the diagnosis of conditions caused by either large genes or multiple genes. And the added benefit of it is that it allowed for unexpected diagnoses. So if a clinical geneticist said, I think this person has X syndrome, we could then interrogate the exome for mutations in those genes. And if negative, we could broaden our search to a broader family of genes of related disorders. And in fact, we showed that that happened in a number of occasions. And when we went back to the clinical geneticists, they said, yes, actually, this fits really nicely with what this person has in hindsight, but we didn't recognize it at the time. The other thing is, at that point, we were at the peak of gene discovery. And so if a person was negative in mutations in known genes, we had the ability to go back and agnostically and reinterrogate the data for gene discovery as well, too. Given my background that I've already told you about with NIH, I also wanted to make sure there was a psychosocial component to my PhD. So my third supervisor for my PhD was Bettina Meiser, who's a psychosocial researcher at UNSW. And that component involved qualitative interviews in a large Aboriginal Australian family with a connective tissue disorder called Marfan syndrome. I could tell you all about that in a separate talk, but it generated really, really rich data. And I finally got around to writing that up, and it's just last month been accepted in Frontiers in Genetics. So I'm really pleased about that. Now, I'm conscious about the fact that when I tell people about my research, that it sounds very broad and not always connected, as it were. So I just wanted to go through with you and explain how I kind of see it all coming together. So I've done a lot of research on genetic conditions, assessing the psychosocial and also the logistical challenges of those conditions. It has produced an amazing diversity of rich data, but recurrent themes that have come up in all of these have been uncertainty. So uncertainty about what my child has for those who don't have a diagnosis, and more often than not, uncertainty about the prognosis and what the future holds for their child. The other theme that comes up is the importance of a diagnosis, both from a psychological point of view, what caused it. It wasn't me standing too close to the microwave when I was pregnant, etc. And also from a practical point of view, I can reach out to other people whose children have the same condition, and I can, and I have a label that will help me to get resources, etc. In terms of the genetic testing side of things, as I said, I looked at how we could better educate patients for genetic testing, but then once they had genetic testing, assessing the impact of that on the individual and on the family in terms of relationships and communication, what impact it had on their health behaviors and their risk perception, and what impact it had on their psychological well-being. And again, my genetic research was focused towards the patient. So looking for new genes for disorders, proving that whole exome sequencing was good for diagnostic purposes, was all aimed at increasing the diagnostic rate for families and thereby speaking to that importance of a diagnosis. And also looking for modifiers goes to the fact that one of the things that frustrates a lot of families is the phenotypic variability within the family. Why is Mary quite mildly affected while Joe has been quite severely affected? And so being able to identify modifiers perhaps allows us to limit a little bit of the uncertainty around that too. So one of the areas that I wanted to focus on a little bit more is clinician education. So as the demand for genetic testing is increasing and the number of genetic tests increases, more and more non-genetic physicians and clinicians are going to be educating patients about this. And so it's really important that they be equipped to do that. And so that feeds quite nicely into my current research program, which is focusing on mainstreaming genetic testing for melanoma into dermatology practices. So in brief, and again, I could talk about this in more detail, but in brief, this involves randomizing clients to get pre and post-test counseling from either a genetic counselor or a trained dermatologist. And our hypothesis is that consumer satisfaction and psychological outcomes will be comparable whether genetic testing is offered by a dermatologist or by a genetic counselor. Now, this study is led by our PhD student, Claire Primiero. This is her baby, as it were, and a substantial portion of her PhD, and she's doing a phenomenal job on this. Ably helped by Betsy Peach, who's a genetic counselor in our group, who has been incredibly important in actually bringing this to pass. Peter has got to be the greatest champion that there is for this cause, and as the Americans say, he has well and truly drunk the Kool-Aid. So he's a big believer in the value of genetic testing and the importance of upskilling dermatologists in this area, which is fantastic. And we have 3 other physicians who are also involved in this study who will provide genetic counseling as part of it as well. So Betsy led the development of the educational materials for the dermatologist. So we start off with a brief didactic session, giving everybody a background that they need in terms of the relevance of the genetics of melanoma. And then they observe a mock genetic counseling session. After that, they observe a real genetic counseling session led by Betsy. And then they do a genetic counseling session themselves, observed by Betsy. And at the end of that session, they debrief about the session and how they feel it went, etc. And if they need any further additional help or assistance, she provides it at that stage. And then from that point onwards, they conduct all of the pre-test counseling sessions on their own. And if the consumer wants to proceed with genetic testing, then a saliva sample is collected at that stage and sent off for testing, and the results come back 4 weeks later. And then the same training process is then initiated for the post-test counseling session. And one of the brilliant parts of this is that we've paired the educational resources with visual counseling aids to kind of prompt the dermatologist as they're doing the pre-test counseling session with people. And they have a checklist and they have an SOP as well too, to help to support them in that process. The other part of Claire's PhD is evaluating whether or not genetic testing and counseling can shift genetic fatalism, which Paul mentioned at the start. And for those of you who don't know, genetic fatalism is the belief that the genetic contributions to a disease override all other factors, such as behavior and the environment. And this is an excellent cartoon that was done by Danica Wadey, who was an executive assistant to Peter for a while and a talented artist. And what this cartoon shows you is the fact that genetic fatalism is not just the belief that if I get the gene, I'm doomed to get the condition, but also the fact that if I don't get the gene, I'm scot-free and I don't have to worry about it anymore. And so it's both of these factors that we'll be evaluating when people test positive and test negative in our study. So just as I'm interested in mainstreaming genetic testing for melanoma in dermatology, I'm also mindful of the other areas in which we can mainstream genetic testing, one of which is pharmacogenomics. So I am an associate supervisor for Priya Iyer, who is doing her PhD in pharmacogenomics, and her principal supervisor is Chris Freeman, and Adam Lacassis, her other associate supervisor. And the project aims to explore the feasibility of incorporating pharmacogenomics into primary care. So looking at GPs and pharmacists' attitudes, their comfort, their knowledge, their competence in doing this, and identifying any barriers and enablers to the process, discussing matters with all the key stakeholders involved. And at the end, Priya hopes to produce a best practice guide for how we might do this, incorporating pharmacogenomics for the management of chronic pain in primary care. So obviously chronic pain is a really good pilot test disease to do that with. So coming back to kind of filling in the gaps in my process, I am going to continue doing gene discovery in my melanoma research. So Betsy Peach was hired by Peter to take detailed pedigrees or family histories from all of our high-risk individuals that were part of the Brisbane Nevis morphology study and the exome sequencing study that he and Rick Sturm did. And in doing that, we recognized the fact that there was a lot of families with what I would classify as Mendelian autosomal dominant melanoma, some of whom have mutations in known genes, but not all of whom. And so a classic example is a pedigree like this, who don't have any mutations in any of the known genes. And so therefore, Bridget Betts Stablian and I put in a grant application to the ASSC, which we were fortunate to get, which allowed us to do whole exome sequencing in about 4 or 5 families like this in an effort to identify new genes. We were going to be doing this face-to-face initially, but obviously COVID had other ideas. And so Betsy Peach is helping us to collect these samples and consents via the mail, etc. And so hopefully we'll be able to put these on for whole exome sequencing shortly and identify a new gene. Going back again to patient education, if we're going to be mainstreaming genetic testing, it's really important that we have as many resources as possible for patient education. And so the year after I got my PhD, I scripted and developed a consent video for patients at the PAH who were having whole exome sequencing done of their tumor and their germline in order to characterize the molecular profile of their tumor. It included all the information that I thought people needed and that I would cover if I was consenting them for the process, and also talks about the risks and benefits of the different types of results that would be generated to that. I produced 2 different versions of the video, one aimed at a general audience and one tailored for an Indigenous Australian audience. And I recently got ethics approval to test that in a clinical cohort at the PA, and also to test it in support groups like Pink Hope and Lynch Syndrome Australia. So I'll have a genetic counselling student who's helping me with testing it with the support groups, and there's a nurse in the genetics group who's going to help me test it in the clinical cohort. Throughout my clinical experience and my research experience, the topic of genetic discrimination has come up time and time again. And it's a subject that I feel really passionately about and have spoken about a number of times of giving talks at different stages. And so I kind of got a bit of a reputation in that area. And so when Paul Lacasse was forming the Australian Non-Discrimination Working Group, he invited me to be a member of that. And it's a really important topic because clinical geneticists and genetic counselors have repeatedly reported that the fear of genetic discrimination deters clients from proceeding with genetic testing, even for medically actionable variants, where doing so would reduce the morbidity and mortality of those conditions. And so as a working group, we looked for various different ways to try and get some sort of protection for people from genetic discrimination. So when the Australian Joint Parliamentary Committee on Corporations and Financial Services held an inquiry into the life insurance industry in 2016-2018, we made a submission and we presented evidence of research demonstrating genetic discrimination, and also how the fear of it is prohibiting people from having genetic testing and also from participating in research. Thankfully, the inquiry concluded by recommending a moratorium to prohibit any life insurers from using the outcome of predictive genetic tests in at least the medium term, and they emphasized that they thought this should happen as a matter of some urgency. So not that long after that, the Financial Services Council introduced an industry-led moratorium. And I emphasize industry-led moratorium because most of the moratoriums in other countries like the UK and Canada and various other countries were government-led. So this is the first industry-led moratorium, and it became effective on the 1st of July of last year. And we weren't sure how effective an industry-led moratorium would be. And so we put in an MRFF genomics application to evaluate the impact of the moratorium and the efficacy of it in achieving its goals. And our hope is that the results of this research will inform the 2022 review that the FSC has in mind in deciding how to proceed with this. So we were lucky in getting that grant, and so that research. So this is the, these are all of the individuals that we will be engaging with as a result of this grant. So all of the key stakeholders: genetic health professionals, patients and consumers, genetic researchers, and also the insurance industry, doing various types of qualitative and quantitative work to capture people's perceptions about the potential value and efficacy of that. The first component of that is a dissemination analysis. So we're keen to see how well the news of the moratorium was spread throughout the media, etc. And so we decided to do a Twitter analysis on that. And Courtney Wallingford is a Masters of Genetic Counselling student at University of Technology, Sydney, that I'm supervising, and she's leading that. And the data was actually extracted by Sally Shrapnel and her PhD student, Amir Ramil. And so we're in the process of analyzing that data right now, but it seems like there was very, very little dissemination that happened. So we're exploring why that might be relative to other moratoriums. An additional component that I'm interested in personally, because I'm a member of the Metro South HREC, is how HRECs evaluate genetics applications. So in my experience being a member of the HREC, the members of the committee weren't quite as confident in reviewing genetics applications as they were in reviewing other applications. And so I wanted to do a research project to explore whether this was true across the board, and if so, was there room to develop new educational resources to empower them. So the aim of this study was to assess HREC's members' confidence in their ability, assess whether the nature and utility of resources that they currently use are effective, etc., and identify whether there's a perceived need for additional resources. And this project is the master's students Ryan Pizar's project from the University of Technology, Sydney. Again, he's a genetic counselling master's student, and his supervisor at UTS is Chris Jacobs. And so this project is being done with Jackie Boyle as well too, who's a genetic counsellor in Sydney, is also supervising him. And we've administered this questionnaire, and we've had 145 responses so far. And to make a long story short, we found that any type of genetic education is strongly associated with genetic confidence. And even a modest amount, which is important as we think about future interventions. What was also true, which is consistent with what I observed, is that people are significantly more dependent on their HREC members in the evaluation of genetics applications than they are in the evaluation of non-genetics applications. And so finally, a lot of my research has been focused on genetic testing for focusing on the individual and on their family, etc. But we're in an era now where people are recognizing the benefit of genetic testing for complex disorders and recognizing the role that genetics might play in that. So we're looking at a much wider population and the value of what we call polygenic risk scores. So for those of you who don't know, polygenic risk scores are basically an aggregate of odds ratios from different risk alleles identified through genome-wide association studies. They're put together in a complex algorithm that's slightly different depending on who's doing the analysis. And rare variants are variants that occur with a minor allele frequency, generally of less than 2% in known risk genes. And the hope is that between polygenic risk scores and rare variant risk scores and Mendelian results, that we might be able to capture that genetic contribution to disease. And in melanoma, this is particularly relevant because twin studies have estimated melanoma heritability to be 55%. And yet mutations in high penetrance genes account for only about 22% of all melanoma prone families. So there's that missing heritability there. And what we know from a variety of other diseases, and Tatiane will speak to this in more depth and detail, is that in high-risk individuals with a strong history who are negative from mutations in known Mendelian genes, they tend to have a high polygenic risk score. So we plan to return the polygenic risk score to our high-risk cohort to assess the impact on their risk perception, their psychological well-being, and any SON-related behaviors. And as I said, Tatiane is going to lead that. We'd also at some point like to include data that Rick Sturm and Darren Smith have been generating through their analysis of whole exome sequencing data, looking at rare variants, and generate a holistic risk score. Peter and I are both really keen that all of these silos come together to the maximum benefit of the patient. So that we take all of the clinical data that we've been gathering, that we somehow integrate with that the imaging data that is becoming increasingly automated, and this is one of Peter's goals long term. And we take all of that data and integrate it with genetic data to generate a holistic risk score for people. And then we triage them and make sure that they get the appropriate level of surveillance depending on their risk, etc. Thank you very much for your time today. Sorry about the technical snafus at the start. There's a lot of people to thank that I won't mention all individually, but I would like to acknowledge my DRC colleagues and obviously especially Peter for his mentorship and support as I get myself established in his research group. So thank you all. So let me go ahead and stop that share. And then I would like to go ahead and introduce you to Tatiane. So Tatiane, as I mentioned, she did her undergraduate at ANU and she did her Masters of Genetic Counselling at Griffith University. And she completed a placement at NIH assessing the impact of genetic testing at that stage. That really kind of sparked in Tatiane a love of genetic research. And so when she finished her Masters of Genetic Counselling, she went on to do a PhD in Sydney with Bettina Meiser there, looking at the polygenic risk score and returning that information to women at increased risk for breast cancer. And she's now returned to Brisbane last year, yay us, and I will let her, won't take up any more time, I'll let her talk about her research. So Tatiane, whenever you're ready, I'll hand over to you.
Thank you, Aideen. That was a really nice segue into what I'll be speaking about. Let me just share my screen. Excellent. Is that working?
That looks perfect, huh?
Great. Okay, so as Aideen mentioned, a lot of my work, mostly for my PhD, has been focusing on looking at polygenic risk scores and how we can implement that in clinical practice. So just give you a brief outline of what I'll be speaking about today is I'm going to start by giving off a very brief overview of what polygenic risk scores, or PRS, what they are. And then I'm going to focus a little bit on just looking at breast cancer risk, because that has been what my research today has been mostly looking into. I'll share some of the findings from my PhD and then looking at how we can implement this in different settings. So just to start with a very brief overview of PRS. So I think a lot of people here would have seen this type of graph where we're looking at allele frequency in the population and then the effect size associated with that allele. And as we know, those who tend to have a larger effect size tend to be rarer in the population. And then those who are more common tend to have a lower effect. As we know with in genetics, in clinical genetics, our work has really been focusing up here. So these are things like pathogenic variants in BRCA1 and 2 genes. This has been really the bread and butter of genetics, clinical genetics work. We see patients with high-risk families who have mutations in these genes. But what we know, as Aideen said earlier, is that these account for a small percentage of disease in the population. And over time, with improvements in genomics, we've been able to then identify variants that are associated with a lower risk through genome-wide association studies that individually have a very minimal impact on the overall level of disease. But then through some additional research, polygenic risk scores, or PRSs, have been developed. And a very basic, I guess, overview of it is that what we're looking at is the cumulative effect of these variants that then is associated with an increased risk. There's different ways of calculating PRS, looking at the effect sizes of them and the number of SNPs that's included. There's a lot of discussion in the literature about the P threshold and which variants to include, and that's some of the work that's still going on in that area. I like to show this picture here. It's one of the images we use in clinical practice. So this was developed by Janine Austin and Holly PA, focusing primarily on psychiatric illnesses, but we've adapted this to other settings. And the way we explain this to patients is looking at your jar. It depends on how many of these beads you have. So people who have a bigger amount of these little beads have a higher risk of developing the disease compared to then somebody whose jar is a little bit more empty. And then you can use this concept to talk about lifestyle. So potentially you might have a different color balls, which are your lifestyle factors that then can increase your risk until you reach the top here where you develop the disorder. Over the last couple of years, there has just been an increasing number of studies looking at PRSs for common complex disorders, from heart disease, from breast cancer, with melanoma, psychiatric disorders, diabetes. There's just been ever increasing amount of studies looking into this. There's been a lot of work coming out of UQ and also Melbourne as well, looking at this applications of polygenic risk scores and trying to develop this work. But I guess there's still a lot of work between developing a PRS and actually having it implemented in clinical practice. And I've been trying to think this through a bit more and thinking a bit more analytical about it and figure out what are the steps we need to achieve to make this actually something that can be used in clinic. And the way I think about it is looking at, firstly, the analytical and clinical validity. And that's things like the GWAS studies and identifying variants that are associated with disease risk. Because in reality, we've only identified a minority of these variants. So giving an example of breast cancer, I think there's only been 313 variants that have been identified at the GWAS level of significance, but we expect it to be, potentially thousands of them, and there's still a lot of work in that area. There's work going on about how accurate our PRS is, what is the best performing PRS, and something that's very important is validating it across different ethnicities, because a lot of this work has just been done in people from European ancestries, and there's research showing that it doesn't work the same way, or I guess the validation across different populations isn't actually quite up to where we would want to be able to implement this in clinical practice. There are still questions around, yes, we might be able to develop a PRS that is accurate, but does it actually improve clinical outcomes over what we already have? And does it reduce healthcare costs? So there's still a lot of questions around that. And the area where I mostly focus on is this side here about implementation. And that's things like workforce training. So, we have clinicians, including in genetic services and looking at other specialties and GPs that will need to be upskilled in how to interpret and provide this information. We need to provide them with clinical resources. How do we then communicate this new type of genetic information to patients? And then what are the psychological and behavioral outcomes associated with that? To focus a little bit more specifically just on breast cancer PRS, from speaking with a lot of clinicians and through my last couple of years doing research in this area, there was a lot of knowledge gap in terms of how clinicians actually interpret this information, and were able to read studies and understand this type of research. So recently we published a review on the topic, which really I wrote it thinking of the clinicians and the type of questions that I had been asked along the way. And it focused primarily on GWAS and variant discoveries, gives a history of breast cancer polygenic risk scores, how it can be used for risk stratification, and then looking at the limitations and the clinical applications. Briefly, when we're looking at breast cancer, I would strongly argue that breast cancer is probably one of the most well-researched diseases in terms of PRS. You know, compared to some other complex disorders, they're just miles ahead in terms of GWAS data and PRSs and prospective studies even, and to the point that I'll show you there's actually clinical testing now available. So what the research has consistently shown is that PRS is associated with a high PRS is associated with an increased risk of breast cancer. It's also associated with an increased risk of early onset breast cancer. And this article here by Madhavat showed that really nicely. Is what they looked at is the different percentile distributions of polygenic risk scores in the population. And these people up here who were in the top 1% of PRS actually did cross the threshold for population screening. So this red line here is associated with age, your risk at age 50, where you generally start population screening. They actually crossed that threshold here at in the early 30s, compared to people who are in the lowest 1 percentile who never even crossed that threshold. And the other thing that's been shown as well is a risk for contralateral disease. So this study here by Sawyer in 2012 showed this really nicely, looking at risk for developing contralateral disease or contralateral breast cancer was associated with your genetics. So if you're looking at here for people who have a pathogenic variant in BRCA1 or 2 that have the highest cumulative incidence of developing contralateral disease, followed by those with a high PRS, and then those here with a low PRS. The thing with breast cancer is that there's actually now prospective studies emerging, and they have been replicating a lot of the retrospective data, which is further supporting the clinical potential for this information. And when we're looking at PRS, I think as well, a lot of people get confused about the applications. It's not just giving everybody in the population the same information and expect the same outcomes. There are lots of different uses to it. So for breast cancer, one of the uses in the familial clinics, so in the clinical genetic services, where we can use to stratify those families, where we look at the pedigree, we know that there is a high risk in that family, but we haven't been able to identify a monogenic or a single genetic cause. We can use PRS to stratify risk in those families. There's also studies showing that PRS can moderate single genes. So people with BRCA1 or 2 mutations have different levels of risk depending on their PRS. Then with people who have a personal history of breast cancer, that information can be used to inform treatment decisions, particularly whether they continue, they have risk reducing surgery or more conservative approach based on whether or not they have a high or low PRS. And then with the unaffected group of women, having personalizing population screening programs and then guiding risk reducing strategies, things like medication that can reduce your risk, surgery or more intensive screening. And then lastly, there's now commercial testing available. And when I say commercial testing, it's not, you know, your direct consumer testing. These are well-established genetic testing companies like Embry, Myriad, Sophia. They all now provide a clinical test for breast cancer, but the test is currently limited to those of European ancestry, people who have a high-risk family history and have an uninformative result from genetic testing, so they don't have a BRCA1 or 2 mutation. So it's a specific group of people who can access this test. So I'll just talk a little bit now about some of the studies that I did for my PhD, looking specifically at breast cancer PRS and some of the outcomes associated with providing that information. So the reason why we did this study is we did a systematic review looking to see what data is out there in terms of how people respond to this information. And we had to broaden the systematic review because there was just really not much out there. So we looked at a whole range of outcomes to genomic testing. We looked at whole genome and exome panel testing, SNPs, and somatic testing. From that, there were only 5 studies that looked at outcomes associated with PRS across different cancers. There was very limited evidence of adverse psychological outcomes among those studies, but they were very limited in the sense that most of them were qualitative. There was only one study that had a longitudinal format or design, and that longest follow-up was up to 3 months. So from that, we've gone on to develop the Variants in Practice Psychosocial Study, or the VIPS. And within that study, I ended up developing 3 different streams. So the first one was looking at clinician training. So this was brand new information for clinicians. We had 6 genetic services involved in the study, but then we needed to train the clinicians to how to provide this information. So we've held 1 workshop at the start of the study, and then I developed another one late last year that was attended by 80 clinicians from across the country on how to use this information. So I've done a lot of thinking about what do clinicians need to know, because obviously everyone's time poor and they don't want to know, they don't need to be given all this information about GWAS studies and things like that. They just want to know, what do I need to tell patients and what do I need to know to interpret this information. I've also conducted some surveys of clinicians. So we have one of 100 clinicians that really shows the level of confidence within this area is quite low. And it really identify areas where they need additional training. I've also recorded about 65 of the appointments where patients came in to receive their PRS. And now I'm doing an analysis to see what are the key things that we need to provide patients with, the key bits of information. And then lastly, which I'll focus on, is looking primarily at the psychological outcomes and the behavioral outcomes of receiving this information. So to date, we've had a couple of studies published. We've had, our protocol has been published. We looked at one providing the baseline data and what are some of the key factors that are impacted women's decisions to receive this information. And then this other one here that was just accepted about last week. It's a qualitative study looking specifically at their responses to receiving this information. So just a brief overview of the methodology. We had about 400 women from Victoria and Tasmania who received letters in the mail telling them, remember the study, the VIP study you signed up to potentially 6 years ago, we now have data, do you want to come in and receive that information? From that, we had 165 who did receive that information and 144 who then completed a survey 12 months after receiving their results. A really good part of this study is that we also targeted those who declined to receive that information. And the reason for that that's interesting is that it's actually really hard to recruit decliners, because they said, no, I don't want my results. And often we don't know why. And because they've said no, they don't want to be part of any additional studies. So we were able to recruit 43 from that 25 completed a final survey. In terms of the demographics, the mean age was about 52, and about half had attended a familial cancer clinic in the past, had a university degree, a personal history of breast cancer, and then 62 received a result that I classified as PRS plus. And PRS plus had a relative risk of above 1.25 to 3.8, and the PRS minus were a relative risk for breast cancer between 0.21 and 1.2. So the first thing I wanted to mention is that receiving polygenic risk scores had very little impact in genetic testing specific distress. So we use a scale called the MICRA, and the distress component ranges from 0 to 30. So as you can see, the adjusted mean didn't even, it was what, 4? So from 0 to 30, that's quite a low level of distress, but it was still an effect of the polygenic risk score received, with people who received a higher polygenic risk score reporting higher levels of genetic testing specific distress. So even though it's still low risk, there's still a difference there, and something that we need to be mindful of when providing this information, that people may feel a little bit more distressed if they have a higher risk. In comparison as well, we looked at uncertainty associated with genetic testing, and again, it was low level of uncertainty, but those who received a higher polygenic risk scores had higher uncertainty. And this is where, for me, the qualitative, the quotes really helps to be able to interpret the numbers, because there's only so much we can understand from the numbers when we're looking at human emotion and human behavior, and having these quotes really helps to unpack that and gives you a much richer data set. So some of the things that people were uncertain about was level of risk for their children, particularly their daughters, and there was a lot of questions about insurance and legal implications. We actually found that about 50% of people reported concerns about receiving their results, given the implications of insurance, and they were really unsure about that. The other thing I wanted to mention is that people had very low levels of decisional regret, and we measured regret about receiving results and regret about not receiving that information. And about 60% of the receivers reported no regret at all. and the remainder had minimal regret. But what we found is there was quite a big difference in regret between those who received the results and those who did not. Those who did not receive the results reported nearly 3 times as more regret about not receiving that information. And that's interesting because we like to think of consent as a one-off approach. You know, you say yes or no now, and that's it. But that's not really the case. And there's other reasons why people might say no. And what we found, there were people who were concerned about the emotional impact of that information. So for example, as this person said, I'm concerned that receiving a positive result could lead to a kind of self-fulfilling prophecy. I will somehow subconsciously expect to get breast cancer and my body will respond accordingly. I do want to know my result, but this subconscious uncertainty does worry me. And really goes to highlight that people have some concerns about receiving this information. And these are areas where we can actually develop some interventions to help them support making that decision and making decisions that they're happy with. And lastly, just wanted to very briefly touch on implementing PRS in other settings. I think the main point I wanted to get across is that PRS has been looked at in a number of different diseases. And the issue with that is that people like to think that we can provide one document that gives all the different risks for across all different disorders. And I personally don't think that's going to work. The reason being that how people respond to different diseases is really going to depend on what that information is and the disorder. We did a study looking at psychiatric for bipolar polygenic risk scores, and the emotional impact of that, being told that yes, you have bipolar, but you also have a high polygenic risk scores. I did those sessions and you could see the body language and people's wave of relief and feeling like I have a high polygenic risk scores. This isn't my fault. It takes away a lot of the blame, guilt, and fear that people have, knowing that there are things that they can do. In comparison, looking at things like heart disease and type 2 diabetes and melanoma, where there's some really strong lifestyle factors that we can target, that's really going to impact how you provide that information and then the risk management options downstream. And then lastly, as Aideen mentioned, I'm now started working with the Dermatology Research Center. I've been very blessed to be welcomed with open arms in the group, and I'm looking forward to doing research now, looking into melanoma. And I think melanoma is another great disease to look at PRS because you can really provide that holistic level of risk, as Aideen mentioned, where we're looking at your monogenic, your polygenic, you're looking at your lifestyle factors, phenotypic factors, to give people a holistic risk. And then looking at how do people interpret that information and what is the effect of receiving that information. And really, I just wanted to highlight again that implementation research is really important, because without it, we can have a perfect PRS, but people are not going to take up that information and it's not going to be utilized for its full potential. So just to summarize, PRS has the potential to personalize healthcare, but it is very important to conduct implementation research alongside analytical and clinical and validity studies. The reason being, if you don't do that, it's really going to stop that information being taken up to its full potential. There is early evidence that receiving PRS is associated with minimal psychological outcomes, but there's still a long road ahead. We're just starting to see the potential of polygenic risk scores. And then just my acknowledgements. So the UQDI team, who I've started working with recently and have really welcomed me with open arms, and I'm looking forward to doing that melanoma study. And then these were my collaborators during my PhD.
Thank you very much, Tatiane. That's fantastic. And thank you to Aideen for a talk beforehand. We're a little bit short of time, but if you wouldn't mind answering one or two questions, is that okay?
Yeah.
Yeah. So we've got a question from Daniel. Daniel, would you like to ask your question?
Yes. So yeah, so I see that there are people who actually regret receiving their PRS testing. I just wonder if they actually got a higher risk, so that's why they feel like they regret of getting that testing.
No, so the people who mostly regretted were those who didn't receive that information. So we measured regret for those who received it, which most of them said no regret. And then those who did not receive it, we measured regret about not receiving that information. And that's those who had the much higher regret levels.
Okay. And Ranjani, would you like to ask your question?
Yeah, so I found it really interesting, and I wondered whether there's sufficient evidence to influence the screening protocols in Australian mammography, because there must be some women who need a lot of screening and others who don't need much at all.
Yeah.
But, you know, there's always the influence of the environment and the factors in the environment which might change the genetic risk would not be apparent so quickly.
Yes, so we did briefly look at that in our study, but because the cohort of women that we were looking at were already at high risk, they're already having appropriate screening before they started. There is a follow-on study now where people are going to be recruited from, it's a national randomized controlled trial where people who come into the clinic for the first time will get invited into the study, and they'll get followed up by 5 years. And there's lots of different streams in that study, but one of the goals is to look at effect on screening and potential health outcomes. There is also big clinical trials going on overseas looking at exactly that. So there's the Wisdom trial that's looking at effect on screening and also health outcomes. And there's a big study from the UK as well. So those are the questions that are still being looked at.
Thanks.
Okay. Well, thank you very much. Those were really fantastic talks, as I say. And if I think in the interest of time, we probably better wrap up there. But Aideen, we want to stay online for the morning tea, I think.
Yes, please. So if anybody would like to stay, for people who'd like to stay for morning tea, please do so, and we'll have a chat there. Thank you so much, Paul, and well done, Tatiane. I really enjoyed it.